Well, I'm very happy to introduce Haley Burrow. Haley did her undergraduate degree at University of California, Santa Cruz. And then we were lucky enough to recruit her here to Lawrence. I forget what year that was, but it was a while ago. And then Haley finished her master's and then PhD here and working on the Dimensions Project. And then 2 years ago, she went to Oregon, and she's going to tell us what she's been doing in Oregon. One thing that you might not have known about Haley is that Haley likes to climb rocks, cliffs, so this was maybe a source of frustration living here in Kansas, that there was so few rock faces. But I think she's been happy to return to the West where there's more rough surfaces. So.
Thank you, Jim. Well, it's so exciting to see so many familiar faces. And believe it or not, even though I am in a place where I can do more of my favorite recreational activities, I do miss Kansas and Lawrence and everyone there a lot. And I think about it often. So yeah, I really appreciate the opportunity to talk to you all about what I've been up to. So yeah, so my title is kind of like encompassing a little bit of like the main project that I was hired on to do here at U of O, which is this relative influence of fire, climate, and vegetation on fungal distribution, but also kind of more side projects and personal interests in continuing my research interests on fungal functional roles and other things I've been up to in Oregon. So yeah, so this project that I'm on is an NSF project, and there are lots of institutions involved, lots of co-authors listed here, and our work takes place on LTER and neon sites. So it's been really cool to be part of such a big team. So generally, with climate change, we're seeing biodiversity loss. This isn't a surprise to any of us at this point. But with this kind of change and loss in species, it's more important now than ever to understand why species are where they are. And in particular, the motivations of this grant were in that fungi are weird. Like they exist in different life stages in different places. And I mean, in general, we know very little about fungi compared to other kingdoms of organisms. But in particular, this grant was began with the questions of why are we seeing fungi in their asexual versus sexual forms in some places rather than others. So for example, like in eDNA from soil, we can pick up a species of fungus that has never been seen showing mushrooms, like so having sexual reproduction essentially is what the mushroom is indicating. And so the title of this grant is Distributions of Macrofungi, Quantifying Ecosystem and Climate Drivers of Fungal Reproduction. So looking across, in this case, the US, and trying to understand how climate and other factors may be driving these differences. Simultaneously, if you're familiar at all with my previous research, I still am very interested in the roles that fungi play and microbes in general in ecosystem function. So fungi in particular play really important role in decay of things like wood and litter, and then nutrient cycling back into the ecosystem. Their below ground bodies in the mycelium play an important role in soil structure. And they play an important role also in plant productivity, which was a large portion of what some of my dissertation work was exploring. So fungal saprotrophs, like I mentioned, are the main decomposers of organic matter. And so more often than not, this is like wood or litter, pine cones, and recycling those nutrients back into the soil. So here I have a few highlights from iNaturalist observations. A lot of our project is using iNaturalist in conjunction with forays at our sites to both collect specimens and also just take note of where we're seeing fruiting bodies of certain species. So I highlighted kind of a variety of different substrates that these saprotrophs are showing up on. So here we have a pine cone, turkey tail on decaying wood, and then sulfur tufts are actually, they can be saprotrophs, but they can also be pathogens of trees. So I'm interested in this gradient between like functional traits and kind of where fungi are acting as pathogens versus saprotrophs, but it's kind of hard to disentangle. So that's something I think about a lot, especially in this kind of case. Um, and yeah, so their hyphae contribute to soil structure, and they also contribute to the carbon cycling in ecosystems. Maybe if you're familiar with some of the Beaver Lab work, and in particular, if Jim or Laura have talked recently about pathogen dilution in driving plant productivity. This is a lot of what my dissertation work was on. And so we had this paper with Guangzhou Wang where we have this relationship between plant species richness on the x-axis and plant biomass on the y-axis, where as there are more plant species, plants are generally producing more biomass in total. So there's this increase in plant productivity with plant richness. And so using pairwise distance of pathogen specialization in sequencing data, so in soil sequences, we were able to create this metric on the x-axis, which is predicted pathogen dilution, and the complementarity effect, which is a similar metric of plant productivity, is also showing this increase. So pathogens, specialist pathogens differentiating are helping drive this response to plant productivity. And further, in one of my chapters in my dissertation, we looked at foliar pathogens, so above ground, not in the soil, and we saw a similar response across 3 years of sampling. So pathogens can be bad, but they can also play a really important role in ecosystem function. Pyrophilous fungi are, maybe you wouldn't consider them a functional group of fungi, but they are fire-adapted fungi. So they are the few fungi that are left after the high disturbance of fire, and they play a really important role in sequestering this immediate flush of nitrogen post-fire, again, stabilizing the soil, and ecosystem recovery. And I think we're still kind of in the early stages of understanding their role in these post-fire systems. So here are a few kind of common species that we see out here post-fire. So on the top, you can see a cup fungus, Pizizomycete, and then morels, which hopefully we've all found. I found my first morels in Kansas by the field station. So, and this really cool orange peel fungus or orange pore fungus that's down at this Klamath Siskiyou forest here in Southern Oregon, south of where I am at. And then the last group that I'll talk about is ectomycorrhizal fungi. So being in the Beaver Schultz lab in grad school, I learned a lot about arbuscular mycorrhizal fungi, and it's been kind of cool to learn more about ectomycorrhizal fungi, being in a place where there's just so many, and they really are really important here, especially in our Douglas fir dominated forests in Oregon. And so ectomycorrhizal fungi help trees with nitrogen and phosphorus uptake amongst other nutrients. including water, tree access to water. There's evidence that they protect against pathogens. And again, like all other fungi, they contribute to soil structure and the carbon cycle. So I highlighted here a few maybe more commonly known or more frequently seen fruiting bodies of ectomycorrhizal fungi that we see here. So amanitas and rustulas. But then on the right, we have at the top chanterelles and on the bottom boletes. And so, yes, there are some tasty ectomycorrhizal fungi, but there are also some ones that we don't want to eat. So it's a fun side note. Right. So because all of these different groups of fungi have such dependence on plant substrates, we kind of expect that like differences in the plant hosts available are going to drive differences in who's there, like which fungi are there. So in conifer forests, we're going to see more ectomycorrhizal fungi, like the rustulas here. And in like the oak litter, we're going to see fungi that maybe specialize on those plant tissues more so than other plant tissues, like in grasslands. And then pathogens are, they're specialist pathogens for particular groups of plants as well. Is everyone hearing me okay? I just saw in the chat that maybe the sound is not great. Yeah? Everyone good?
Yes.
I think it's fine.
Okay. Okay, great. Thank you. So kind of the main questions that I'm gonna touch on here today are, to what extent do these groups, pathogens, saprotrophs, and ectomycorrhizal communities differ across vegetation types, given this expectation that there's specialization, differentiation across different kind of vegetation types or ecosystems? And then are there similarities between these vegetation types across ecological regions of the US, despite climate differences, fire history, and just like geographical differences? So in order to touch on these questions, we have sites across the US, and here's an image of the general climate gradient across the US. And so this is incorporating both temperature and precipitation patterns. And we have sites at each of the star locations. So down in the taiga, or up, I guess, up in the taiga in Alaska. It's kind of weird where I put Alaska here. That doesn't really make sense, but the Pacific Northwest in Oregon, we have a site in Arizona, the Rockies in Colorado. We have a site in Kansa, which is the prairie site. Great Lakes up in Minnesota. In the Northeast, we have a site in Massachusetts, and in the Southeast, we have a site in Florida. And so we have estimates from the Prison Climate Group at Oregon State University with 30-year averages of temperature and precipitation across all of these sites. And so I have them plotted here just to show the range, first of all, of temperature and precipitation across sites, as well as the differences in when things are peaking versus dipping off. So we would expect, I mean, fungi are highly dependent on temperature and moisture availability. So we would expect these differences to be affecting which fungi are where. So here's again a map with those sites. They're, like I mentioned at the start of the talk, neon and LTER sites. And at each site, there were 3 vegetation types that were selected. So we have pine, grassland, and oak woodland. And then within each, we tried to find a burned within 5 years plot and a burned never or greater than 30 years ago plot. And at each plot, there are 3 transects with that are 20 meters apart and 9 subplots. So at each subplot is where we sampled. And by we, I mean mostly my collaborators, because I was brought onto this project towards the end. And I got to help with the last sampling period when I first started, but just in the Pacific Northwest site. So it really put in perspective how much effort was going in across all of these sites across the 2 years that we sampled. And so here's a nice kind of diagram breaking down there's the 3 habitat types, so conifer, oak woodland, and grassland, and we have burned and unburned. And then what we sampled is in the middle here. So we sampled sporocarps via forays at each site. Endophytes from leaf tissue that were cultured and then Sanger sequenced. Soil and litter via cores. And then there were spore traps that collected spores from the air. And this was, for the most part, at all 9 subplots for each of these plots. And then, depending on the sampling type, we did either PacBio or Illumina sequencing. And today, I'm going to talk to you about the soil and litter data because we're kind of in the process of knitting together all of these sequencing data, which a grad student here at U of O is working on the bioinformatics for. And it's quite remarkable, but it's quite an undertaking as well. But first, I wanted to show some fun highlights from the Kanza site, because I figured you all might want to know. I know some of you were part of the foray that they did in Kanza, and that you likely go visit Kanza often. So wanted to give a shout to that. So the macrofungi collected in Kanza, so there were 0 collections at the site beforehand, and we now have 180. And about a third, almost half, have been verified with PacBio sequences. And about a third are new to Kansas. So this is based on herbarium records that my supervisor, Biddy Roy, has been going through each and every PacBio sequencing run and confirming its identity to the spore or the macrofungi, the collection. There is one new species new to the US, to our knowledge, which is this one pictured here, Ryopa metamorphosa. And you can see it here in both its asexual and sexual stage. So on the right is its fruiting body, its reproductive sexual stage, versus on the left is it's just growing in the wood asexually. There are a lot of crusts at Kanza, and they're really cool, and crusts are just generally very easily overlooked. So I highly recommend, if you go to Kanza and you're just hanging out, flip some things over, look in the nitty-gritty areas that you may not just see just while hiking, because there are some really interesting crust fungi. And in particular, one here is found by Robert Menning, who I worked with when I was there in grad school. So that was kind of fun to see, a familiar name in the data set. There are also quite a few shelf fungi. And from what I've heard of this foray, it was really quite dry that day. So I think it makes sense that a lot of the fungi that we're seeing were quite dry. And shelf fungi are, they do become more hydrated when it is wetter, but they also stick around in dry conditions. So quite a diversity of shelf fungi. And then there were some mushroom, just like typical mushroom forming fungi as well. And so this observation by Thomas Koffel, who is a postdoc when I was at KU as well. So it's, again, fun to see familiar names. Okay, so circling back to the questions in the project. So we have these three different vegetation types, the oak, grassland, and conifer. And so the hypotheses that I have with this soil and litter data is that fungal functional groups will differ across these different vegetation types and further across the ecoregions. And that saprotrophs, pyrophiles, and ectomycorrhizae will be particularly sensitive to pH and C to N nutrient availability just due to their inherent known contribution to nutrient cycling in ecosystems. All fungi are typically thought to be pretty pH sensitive, so we also just generally expected pH to be affecting all fungi. And then we have a vast range of what burning and fire looks like across these sites. So in most cases, they're prescribed fires, but conifer forests are going to burn at a much higher severity than grasslands, for example. And even across all of our sites, whether they were prescribed burns and how frequently they burned was kind of variable. So we're still kind of working on differentiating between those different like elements of burning. But generally, due to these differences, we expected there to be a differential response of the guilds to the fire treatments at each ecoregion. And in particular, we expected ectomycorrhizae to be affected by fire in the oak and conifer vegetation types, and that pyrophilous fungi would be most affected in conifer sites. Though there are pyrophilous fungi in kind of all vegetation types as well. Right, so as I mentioned before, we had sampled, or there were soil cores sampled across all of these sites at all of these subplots. And there were 4 sampling periods. So there was one in the spring and the fall of both 2022 and 2023. Prior to sequencing, soil and litter were separated. And then PCR of the ITS1 region of ribosomal DNA was amplified and then sequenced with Illumina sequencing. In spring of 22, so the first sampling, there were pH, carbon, and nitrogen measurements taken from both the soil and the litter separately. And then the bioinformatics and ASV table were produced. And then I used a database called Fungal Traits to filter out putative saprotrophs, putative pathogens, ectomycorrhizal fungi. And then we've been working on a list of pyrophilous fungi to basically manually filter out taxons that are known to be pyrophilous. And we're still kind of working on that. So maybe some of the pyrophilous fungal data isn't quite what it will be yet. I think it's on the smaller side right now, and I think we'll be expanding on it a bit soon here. So kind of unsurprisingly, the biggest effect, biggest main effect on each of these groups was the ecoregion. So on the x-axis here, we have ecoregion, and simultaneously looking at these figures, it's hard to really see major differences between the different ecoregions. And so on the y-axis here, we have diversity for each of these groups. And it's likely that these statistical differences are driven by things like weather, climate in general, climate patterns, and particularly other like maybe soil differences and things like this across the sites. So we're hoping to add in more of those factors into these models to hopefully explain some of this variation. Pathogens, ectomycorrhizae, and pyrophilous fungi all had significant main effects between these vegetation types. So On each of these figures, we have the diversity on the y-axis, and we have conifer, grassland, and oak on the x-axis. And you can see, so for pathogens, their diversity were higher in the grassland and oak vegetation types than in the conifer. And ectomycorrhizal fungi were not surprisingly more diverse in conifer and oak woodlands than in grasslands, because that's, most of their hosts are trees. And then pyrophilous fungi were interestingly the most diverse in conifer sites than in grassland and oak sites, which there are accumulating evidence to support that there are more fire-adapted fungi in conifer forests where burns are more severe, and there's also more time between burns on average, typically. I will point out here, though, that the y-axis for ectomycorrhizae and pyrophilous fungi are comparable, but for pathogens, generally their diversity was higher than the ectomycorrhizal or pyrophilous fungi. So just to point out that y-axis starts at 2.2, which is maybe a little, good to know. Right, so moving on to some of the nutrient data. On the x-axis here, we have carbon to nitrogen ratio. And we did a ratio because sometimes the raw carbon and nitrogen measurements are a little hard to interpret, although ratios are sometimes hard to interpret too. So we generally found that ectomycorrhizal fungal diversity decreased with the carbon to nitrogen ratio. And I have this little helper to think through what that even means. So as there's more carbon in comparison to nitrogen, the diversity of ectomycorrhizal fungi decreases, which is interesting. So in high nitrogen conditions, there are more diversity of ectomycorrhizal fungi. And we're just looking at the conifer and oak woodland in this figure because there wasn't a relationship for grassland. For pH now, on the x-axis, we found that pyrophilous and pathogenic fungi, their diversity increased with pH. So on the top, we have pathogens. On the bottom, we have pyrophilous fungi. And again, just a reminder of what that means. So 7 and 8 are kind of neutral, and then 4 is going to be more acidic. So seemingly pathogens and pyrophilous fungi, a higher diversity of these fungi are existing in more neutral soil and litter compared to more acidic conditions. And then with the fire response, we actually haven't found a significant difference in pyrophilous fungi between the burn and unburn treatments. But I'm hoping that once we have that list and subset a little more fleshed out, that we will maybe see some more interesting responses there. But ectomycorrhizal fungal diversity was significantly higher in the unburned treatments than the burn, and this is a main effect. We're thinking that maybe like ectomycorrhizal fungi are just more sensitive to fire, and so it takes longer for them to recover post-fire. And so that was an interesting result to think through as well. Okay, so I just want to summarize some of these things that I just showed. So the greatest variation explained was by ecoregion. And so some of our next steps are to incorporate local weather station data and subplot level humidity and temperature data as well, to kind of hopefully explain some of that variation apart from just the bin of ecoregion, which can really encompass a lot of things. I showed some diversity responses where pathogen diversity was highest in the grass and oak sites, ectomycorrhizal diversity was highest in the conifer and oak sites, and pyrophilus diversity was highest in the conifer sites. We also are doing compositional analyses, so using permanovas, and I just chose to show diversity, but we also are running richness and relative abundance to kind of get at separate sub-questions. So this is just kind of scratching the surface. And then we saw that ectomycorrhizal diversity decreased as carbon to nitrogen ratio increased, and that pyrophilus and pathogenic fungal diversity increased with pH and ranged from kind of acidic to neutral. And then finally, ectomycorrhizal fungal diversity was higher in the unburned sites, which is potentially suggesting that they are sensitive to fire and need time to recover. Right, so as I mentioned, some of our next steps are to use the weather and climate data to assess the contribution to these regional differences. We have community responses, and we're also hoping to have the full data set of all of the compartments, so the sporocarps, the endophytes, and the spores all together to look at overall community responses. And then finally, we're working on some species distribution models for particular taxonomic groups that are more common across the regional distribution. So that'll be really exciting because I think we have a pretty unique opportunity to look at some of these questions. Okay, so that's kind of my work on the CliMush project so far. And then I wanted to talk a little bit about this decomposition experiment that Ellen Ralston pictured here, who is an undergrad in Biddy Roy's lab last year. They just graduated, but they did their undergrad thesis on this decomposition experiment. And I've kind of picked it up to do some sequencing and hopefully write it up into something very interesting. So there were 3 different sites of grasslands that had burn and unburned plots. And at the beginning of the experiment, leaf litter from these grasslands were collected and placed in mesh bags, as you can see on the left-hand side here. A separate part of this is that like half of the bags were sterilized using autoclave, and the other half were not, to see whether that would affect decomposition rates. And we're still kind of trying to understand what what our expectations even were there. So I'm not going to talk about that too much today. Talk mostly about the differences between the burn and unburn. But yeah, so there were enough samples placed at the beginning that bags were removed at 3 months, 6 months, 9 months, and 12 months. And then after removing the last round, I did ITS1 sequencing to look for fungal DNA. And so the hypotheses we have for kind of like the hybrid of both of these data are that decomposition rates and fungal saprotroph diversity will be higher in the unburned sites. Assuming that burn sites have less litter to decompose, so maybe there are fewer fungal saprotrophs, and that potentially saprotrophs are sensitive to burn or fire. And then I was kind of hoping that fungal saprotroph diversity would be correlated with decomposition decline, kind of showing that there is a contribution of fungal saprotrophs to decomposition. And so, oh, right, I guess I didn't mention. So at each time that these bags were collected, the after weight was recorded. So we have the before weight and the after weight to measure the decomposition over time. So here we have on the y-axis that mean weight difference. So from after or yeah, after minus before, or before minus after. Sorry. And then on the x-axis, we have burn and unburned treatments. And we saw a significantly less difference in weight in the unburn. So I guess there was less decomposition, or decomposition was higher in the burn plots. But we saw in the sequencing data with saprotroph diversity on the y-axis, kind of the opposite. There was not a main effect of fire. There was a site by fire interaction. And so we have each of the three sites here, the Eaton, Pisgah, and Spire. And across these two sites, there wasn't a significant difference, but there was in the Eaton site where saprotroph diversity was highest in the unburn plots. So we're still kind of trying to figure out what this all means. And unfortunately, there was not a correlation between decomposition from the litter and the saprotroph diversity. So the next steps for this are to kind of look more at particular taxonomic species of saprotrophs and look for indicator species that are maybe driving the decomposition response that we're seeing with just the bag weights. I also have, compositional analyses to sort through. And I think too, looking at more specifically this chronosequence of removal of bags might be interesting, because we did see an initial response with the bag weights in that first 3-month collection, and then it kind of tapered off later on with the later collections. So we're hoping to explore that a little bit more with this data. In the fall of 2024, last fall, I got to teach mycology at U of O, and it hadn't been taught since 2021. And the previous instructor, Jeff Stone, is retired, but he's in town. So I was able to get some help from him and put together a really awesome course with 20 just like awesome students who are super excited and also very forgiving with me in my first time teaching the class. And there were 6 weekends where we had field trips, and so students had their sporocarp collections that they then brought into the lab to do ID using dichotomous keys. And here you can see a couple of my students with some giant boletes that unfortunately I think ended up being too buggy to eat. And kind of a picture of like the diversity of fungi in general that we found. We got to go to this waterfall one of our field trips, which is just, I guess waterfalls are still impressive to me, even after being here for a year and a half. And it was an amazing chanterelle year. So, I mean, this was just one of our forays, and I wasn't even really trying to look for them. I was just out with the students, and it was hard not to grab them, you know? And then also part of this is that we got to do a microfungal collection where students cultured microfungi from various substrates. And then we used keys from a variety of books to key out particular microfungal species. So it was a really cool experience. I hope that I'll get to do it again this year, but they're still kind of looking into funding, and I feel really lucky that I got to do that. Okay, so this is my thank you slide. And so thank you all for listening. And these are some of my collaborators collecting fieldwork and presenting. So I have a lot of people on the team that I work with to thank. And I put the QR code to our website, the CliMush website, in case you are interested in learning more about the project. And my email's here too, in case you want to ask questions after this or reach out for any reason. Yeah, and I'm happy to take questions.
That was awesome, Haley. So we're people here at KBS. I don't know, I can look at you and call out questions, or people online can type them in. I have a question, Haley, about your site. So how much were you able to deconfound location from your plant community types? Like when you were at Kanza, did you sample the oak forest at Kanza too? Or are we just looking at the grassland at Kanza?
Yeah, so there are, in most cases, each vegetation type represented at every ecoregion.
Okay, cool.
So things like, places like oak woodlands, there's a lot of oak species that are just common across the US. But like we're seeing actually in the spore data that the Great Lakes location up in Minnesota, that their grassland and oak woodland are most similar to Kansas, which makes sense because, the Great Plains. So so there's definitely some yeah, kind of like more overlap in across some ecoregions. But there's more overlap between vegetation types across the US than one might think, actually. Mhm.
Jim, could I ask a related question? Go ahead. It has to do with the sampling on Kanza. Kansas isn't really known for its coniferous forests. And so were you sampling from eastern red cedar areas on Kanza for those samples?
Yeah, I'm pretty sure that that is what we did. There are a couple sites. Um, I'm trying to remember. I don't think Kanza is one, but there were a couple cases where we weren't able to find a community type at some ecoregions. Or like, for example, in Oregon, we have, in most cases, all three vegetation types were under one LTER or neon site. But in Oregon, our oak and grassland are at a completely separate part of Oregon than the conifer site. So,
But it
yeah,
is with the cedar is AM, associated with AM, so it's not,
yes,
so that could explain the big effect of the regional
right.
differences. Yeah.
Yeah, and I think I'm trying to, yeah, I feel like I should probably know this, but I'm actually thinking that maybe we didn't have a conifer site at Convent now because we specifically were looking for pine
Yeah.
forests.
Yeah. Good.
So yeah, I think actually Kansa's the one that we don't, now that I'm thinking about it. Yeah. Yeah, because that's a great point. Red cedar, yeah, wouldn't have ectomycorrhizal fungi.
So Sarah has a question here.
Can you hear me?
Yeah.
Okay. Your relationship with carbon to nitrogen ratios. What is that, the carbon to nitrogen ratio of? Because the numbers are very high for soil. So is it litter?
It was both. So I guess didn't separate out soil and litter. I combined them, um, just like in that plot. And we actually, yeah, we had some questions. Our collaborator who was kind of directing the nutrient analyses had that question too, and was kind of questioning whether our nutrient data were accurate. And unfortunately, we don't really have a way to redo it, or at least with the resources that we have at this point. So yeah, that is something that we have noticed. And that's, I think, part of why we did we used the ratio, because yeah, the carbon numbers were a little all over the place. And I will note, I think we had talked about that the litter difference from the soil varied a lot across sites. So for example, the Alaska site, litter, like there really isn't a difference between litter and soil. It's really just like all, like the soil is very airy, and so it was hard to across sites standardize the separation of the litter in the soil. So I think there was maybe a little bit of limitation in the methodology there. That's worth noting.
Well, thank you, Haley. This was wonderful.
Thank you.
Very interesting.